Micro.
Hi, everybody. Welcome. We'll give folks just another minute or two to filter in here. And as the rest of the attendees filter in, I'll just start us out with a couple of announcements before I introduce our speaker, our first speaker today. So, hi everyone, welcome back to the Long-Term Animal Research Seminar Series. We're glad you could join us. We're going to keep doing this for at least one more semester, and we'll say that every time we start a new semester of talks for perpetuity. So we're glad you're here. In the first seminar of the new semester, we've got two talks today featuring insights into wild mandrills, which I'm really excited about. We'll start with a shorter talk from Nikolaos Smit and then finish with a longer talk from Merci Charpentier. A couple of details before we begin. First, if you're joining us via Zoom, you can submit questions at any time for our speakers via the Q&A tab. And at the end of each talk, we'll start with those questions that have received the most upvotes. And then second, this talk is streaming on YouTube and will be uploaded to YouTube shortly after it's complete. So it's going to be available to anyone who you might know who couldn't join us live and wants to watch it, or if you want to rewatch it 2 or 3 or 10 times, you'll be able to. So I will start by introducing our first speaker today, who is Nikolaos Smit. Nikolaos has received master's degrees both in biology, where he studied acoustic communications in crickets, as well as in algorithms, logic, and discrete mathematics, where he studied network theory, and he's now completing his PhD at the University of Montpellier. Nikolaos is combining those interdisciplinary skill sets to better understand social relationships in wild mandrills, with a focus on heterosexual relationships and sexual conflict. He's just recently returned from the field in the last couple of weeks, so I'm grateful that he could get a talk together and join us today and tell us about his work. I'll turn it over to Nikolaos.
Thank you very much for this presenting. Thank you very much for having us, and Liz and Matthew. And we are happy to start the new semester of these great seminars. So I will share my screen now. So I'm indeed this Nikos guy, and I am working on intersexual relationships with mandrills, and today I will present you my first work on sexual coercion and sexual intimidation in mandrills. So first of all this happens in the space of Mandrillus project, which is based on southern Gabon, as you see on the map. I will now say a few words about mandrills, and after Marie Charpentier, who is my supervisor, along with Elise Houchard, will give more information about our project and the species. So mandrills live in large polygynous groups. There are seasonal breeders. Here we see the months from January to December, and males immigrate mostly in the group in the summer, the European summer, with a peak in July and August, which coinciding with the swollen period, the days, the weeks and months that females are exhibiting sexual swellings. Males compete severely, and as a result, they have evolved sexual characteristics and intense characteristics, such as the color that you probably know, as well as the long canines, which are extremely long for a primate order. Mandrills also exhibit major sexual size dimorphism, with males being 3 to 4 times bigger than females. We know that males aggress females in the species, but we also know, and we observe in the field, that that females use strategies such as avoiding males or creating all-female collisions against males. So these reasons, these things together, made us study first, me for my PhD, the sexual conflict and precisely the sexual coercion in the species. So a few words about sexual conflict. It emerges from the diverging evolutionary interests between the two sexes, which emerges with its turn from the reproductive success, which is in the case of females is limited by the quality of mates, while in the case of males is limited by the quantity of mates. So in this context, males might use aggression in different forms in order to incite females to mate with them, with the aggressor, or prevent them from mating to mate with rival males. So the question here that arises is if male mandrills coerce females, and if this is the case, how, with this, what strategy? In order to study that, we adopted the sexual coercion hypothesis, as per Smit and Smit, which predicts that, first of all, cycling females receive more aggressive assaults by females in other reproductive states. We group I will explain it later, but I can mention already that we group the females 4 reproductive states, which is cycling or swollen females, lactating when they have a dependent infant, pregnant, that I think all of you know what it is, and a default state, which is like not coinciding with any of the previous 3. Second prediction, that this aggression from the males is costly to females, and we studied that through injury patterns. Third and last prediction, that this aggression increases male mating success. For this last third prediction, we split our analysis in 3 distinct scenarios. First of all, for short-term harassment, when copulation, we tested if copulation follows directly a few seconds after aggression. Second of all, mid-term coercion, testing if aggression and copulation occur both in the swollen period, the swollen days of a female. And lastly, we tested for intimidation, in case that aggression and copulation are temporally decoupled, and I will describe in detail in the next slides what this means. So for the first prediction, we see here in this plot, which has a reproductive state with the 4 reproductive states, as I explained them earlier, in the x-axis, and the y-axis, the aggression rate from adult males towards adult females, that indeed swollen females receive significantly more aggression than females in other reproductive states. And here in the precise figures, you can see that's almost 2 times more in comparison to all the other states, pregnant, no swollen, and lactating. So our first prediction is indeed confirmed, with swollen females receiving significantly more aggression than females in other states. In the second prediction now, in a similar graph, which plots the reproductive state on the x-axis against the probability of being injured in the y-axis, we see again that swollen females have higher probability of being injured. Again, we see here that the difference among swollen females and the other 3 states are big. So our second prediction is also partially confirmed, and in order to strengthen this result, so in order to clarify the origin of the injuries of the females, and if these injuries are due to male aggression, we ran a similar model as the one of the first prediction. So here we see what we saw in the first prediction, with swollen females receive significantly more aggression, but this time we tested, and now I move to this graph on the left, if swollen females receive significantly more aggression also from individuals other than adult males, which is not the case, as we see here, that swollen females receive less aggression, for example, than pregnant females. So indeed, our second prediction is confirmed again, because the most likely cause of the female injuries is the male aggression. So let's go now in the third prediction, third and last. In the first scenario of short-term harassment, we used all the focal observations, in case that some of us, we don't know what's a focal observation. It's a fixed time span in which the observer records the sequence of behaviors based on a protocol of a given individual. So in our case, the focal observations last 5 minutes or 300 seconds. So I took all these observations, and I tested if there is aggression during the first half of the focal. In the case that there was indeed an aggression event, let's say in this point here, I tested if there is copulation during the 150 seconds, which coincides, it's equal to the half of the focal again. So if in this time span there is copulation, and for match control cases when there is not aggression during the first half, I tested if there is copulation during the second half. But the case is that they were not correlated, aggression and copulation within the focal observations. So male aggression does not increase the probability of copulation with his victim in the short term. So short-term harassment doesn't likely exist in mandrills. In other two scenarios now, we use a much different time scale. So here we have the arrival of the male that we study at any case in the group this year, because I said they are immigrating into the group. And here we have the onset of the swelling period of the female until its end. So in both other scenarios, the mating window, so the time window where we test if there is copulation, it's the swollen period of the female. So let's go first in the midterm coercion. Here we test if aggression during this same window, this mating window, during the swollen days of the female, is correlated with mating. And as we see also in the graph bottom left, which plots the copulation, the probability of copulation basically, against the aggression rate received from the female during this window, we see that there is not a correlation between aggression and mating. In the last scenario, the intimidation scenario, aggression and mating and copulation are decoupled, and we test if aggression from the arrival of the male in the group until the beginning of the swollen period of the female is correlated with copulation during the swollen period of the female. And indeed here we found a correlation with aggression rate being, as we see in bottom right again, in a similar graph, that the aggression rate of the male during this aggression window this time is is increasing, let's say, along with the copulation probability. So to sum up, we show here that male mandrills use probably long-term, not probably, we show it, sorry, we use a sexual coercion strategy, the long-term one, which is called intimidation, and it's unlikely that they use short-term harassment or mid-term coercion. It was mentioning here that there are only other two, in our knowledge, only other two studies of primates and sexual coercion in primates, which is one in chimpanzees and one in chacma baboons, that they both saw sexual intimidation and not sexual harassment, show only long-term strategies, suggesting that species that form polygynandrous groups and have high sexual size dimorphism, they are likely to exhibit also sexual intimidation. There are of course also studies showing that like the extreme case of sexual coercion, which is the forced copulation in orangutans, but I don't mention that here. And lastly, yeah, the next step of my PhD also is to study the side of the female strategies in this context. That's all. Thank you very much again. And I forgot to thank, of course, my supervisors and all the field assistants and everybody who is involved in Project Mandrillus.
Great, thanks, Nikos. Well, that was really interesting. I have a couple of questions. I know Liz has a question. People can put their questions in the Q&A if they have questions too. So let's start with the question from Liz.
Yeah, that was a really great talk, Nikos. I was wondering if if there's cost to females, especially ones who receive injuries, if they're less likely to conceive.
I mean, if injuries are correlated to probability of conception.
Yeah.
In my I don't know. I don't think it's studied that in mandrills. In my knowledge.
Cool.
I don't know it. Sorry.
No, it's okay. I'm looking forward to hearing more about the female side too. Really nice work.
Okay.
So Patrick Green has a question. He says, although aggression rates from the other group members don't seem to differ across the reproductive cycle, they do seem high overall. So are females being aggressed by other group members at a high rate? And why is that?
In general, I would say that in comparison, for example, with baboons, that mandrills are usually compared, mandrills are more peaceful in this respect. So females are aggressed, yeah, but yeah, the rates of aggression are not very high, I think. So yeah, they're aggressed also by other group members, but yeah. And usually the aggression, the aggression we observe, it's maybe from, whens between females, for example, because in this plot I showed it, there are females and juveniles pulled together. Yeah, there are maybe from high-ranking individuals towards there are top bottom, no top. Yeah.
So my question is about why females swell at all, right? So if the swelling is this attractive, seems to lead to increased aggression from males, right? What are the benefits that make up for those apparent costs, the injury costs and other things?
I'm not sure I understood. Sorry.
So females display these large sexual swellings, right? And so my question is, given that the presence of those swellings seems to induce male aggression, right? Since males aggress when they're swollen, what benefits do females get from swelling that make up for those costs?
Okay. I would say that there is always the context in these cases, in this like complicated social systems of antagonistic evolution. So I cannot reply like directly how, like, I understand the question. Give me a second to think. You know, since we don't have studied female choice yet in mandrills in detail, it's hard to know the benefit of females. I hope I will be able to answer in 1 year or two. But yeah, seems that females have some sort of choice in mandrills. So yeah, there should be other reasons to exhibit their ovulation or their anyway fertility somehow.
Great. Okay, great. Well, thanks, Nikos, very much. I think given the time, we should probably move on to Marie. I'll look forward to the follow-ups and looking at female strategies. Our second speaker today is Dr. Marie Charpentier. Marie completed her PhD in evolutionary biology at the University of Montpellier. She received a Marie Curie Fellowship and worked as a postdoc at Duke University, working with both Susan Alberts and Christine Dray. She then returned to the University of Montpellier as a permanent researcher, and in 2012, she established the Mandrillus project in southern Gabon, the only long-term study of habituated mandrills. She uses a terrifically integrative approach, combining daily behavioral observations along with non-invasive biological sampling and photogrammetry, as well as groundbreaking techniques using AI and other novel biomarkers to understand these animals' social behaviors and their evolution. The project has led to major advances in our understanding of kin recognition and the relationship between parasitism, sociality, and kinship. And so I'm excited to welcome Marie today to hear about her work so far and where it's going in the future. Marie?
Yes, thank you for this very nice introduction. So I'm going to share my screen now. Yes. No. Is it working? Yes? Okay, perfect. So thank you very much. I'm really, really happy to be online with all of you today. And I would like to thank, of course, the committee organizer for inviting me to present my long-term field project in Gabon. I'm sorry, I'm going to read my text because my English is generally better and my accent is generally better when I'm reading. So sorry for that. I hope it will be a kind of alive anyway. So today I will show you a few old results. So I'm sorry if you have already heard them, but also more recent ones that we could not have obtained without being every day or almost every day with a social group of mandrills I'm studying for the past 9 years now. So that's why I partly titled this talk, Long-Term Field Studies May Reveal Unnoticed Details of Social Complexity. So since the beginning of my career as a researcher, I've been particularly interested in the study of the different determinants that influence social relationships in primates. And I'm still trying to answer the question, which individual, social or environmental characteristic impact the formation, the maintenance and the strength of social bonds among individuals in a given social group. So this characteristic may be, for example, how food is distributing in the environment, how individuals are organized in hierarchies, or their genetic relationships, a parameter that I've long focused on, as I will show you a bit later. So before going further into science, let me present you the social group of mandrill I'm studying since early 2012, as Matthew says. As you may know, wild mandrills live in the deep forest of Central Africa, mainly in Gabon, where I have established, and I'm still running a long-term field project in the Lekedi Park here, near the village of Bakumba in the southeast of the country, very close to the Congo border. So something that you may not know is that wild mandrills are almost impossible to habituate in the very dense forests where they live. Luckily, in 2002 and 2006, 2 small groups of captive mandrills were released into the Lekedi Park. Starting as early as 2003, wild males joined the group to reproduce, and currently only 7 old females are born in captivity, representing less than 3% of the population. But these old females allowed in 2012 to habituate the remaining individuals of the group to our continuous presence. So it took my team, and I about a year to be able to follow them the entire day, and a few more years to have them all really well habituated to us. So right now, a team of 8 to 10 persons are daily following these mandrills to collect many types of biological data. And since the end of 2020, we know individually all of the 250 mandrills of the group. And believe me, in this kind of habitat where they live, this is a huge achievement. So some females of the group are equipped with radio collars to find them back when we lose them, which occurs almost every month for a few days. The group is large, as I said, 250 individuals this year. And you can see, for example, a subset of our study group crossing a path here. So we experienced also one episode of fission in 2019 that lasted a few weeks only, but otherwise the group is stable and cohesive. In 2012, I've also set up a field lab where my field assistant and I are doing basic parasite and hormonal analysis on a daily basis too. So we are housed in and have our lab facilities in the park in a large and quite welcoming and fully equipped station, as you can see on this picture. So after this quite long presentation of the site, a bit more about the data we are collecting. So in the dense forests where mandrill lives, it's impossible to count group members when they are not crossing a road like this, which happens from time to time. So we are also daily following their group movements. Here on this map, you have an example of a 6 months occupation of a small part of their home range. We are also making daily censuses and are collecting data on female sexual cycles. And trained field assistants are doing ad libitum and focal observation of social and solitary behaviors. So we have obtained, for example, almost 7,000 hours of focals since the beginning of the project. We are also daily collecting non-invasive fecal and other samples. All these samples are directly processed in our field lab. Coprologies and cortisol analysis are performed in routine on all fecal samples while we are measuring other biological parameters from time to time. And punctually, and until 2015, we have also captured some individuals of the study population, and we are finally performing various protocols depending on our own needs or those of our collaborators. And we have also accumulated a very large photo bank of 30,000 something portraits obtained from 300 different individuals from the group, and for some of them since their birth. So here you can see a small sample of pictures taken on one adult female during a few months. So I will talk about this photo bank a bit later. So all of these data from the field are sent to me every single month. So I'm cleaning them 2 or 3 times a year when I have time. And then I incorporating all of the relevant information into a user-friendly database, which will be hopefully available soon. So if you want to learn a bit more about the project, you can visit our website at this address. So now a glimpse on what we are currently doing with this long-term data. So I'm co-supervising 4 PhD students right now in collaboration with Élise Huchard, Julien Renou, and Peter Kapler, who you probably know. So Nikos, that you have just heard, is working, as he said, on heterosexual relationships in mandrills and on males and females reproductive conflicts using the long-term behavioral data available. Axel here is studying sibling rivalry, both in baboons and in mandrills, again, using this long-term behavioral data from the project, but also from the Tsaobis baboons research project in Namibia. Sonia is working on the evolution of beauty using the long-term photo bank collected on mandrills and artificial intelligence methods with Julian. And finally, Berta, who is just starting her PhD, will work on parental care in mandrills using data she has herself been collecting for the past 2 years and from long-term monitoring on mandrills infants. So aside from this students project, I have myself developed 3 lines of research. So a first one in health ecology with 2 projects that have been or will be hopefully funded soon. One on the immune theory of sleep and the other one on the relationship between the social microbiome and individual fitness. So this latter project will be done in collaboration with Alice Baniel, who is currently a postdoc at the Arizona State University with Noah Snyder-Mackler and Amy Liu. So I'm still developing researches on the influence of parasitism on social behavior, a project that started with the PhD of Clémence Poirot, who is now in postdoc in Peter Skapler's group. And last but not least, since my PhD, I spent quite a lot of time studying kin selection and how kinship structures social relationships in primates. And this is the research line I will talk about today. So a bit of background on the fascinating biological model I'm studying for the past 20 years now. So Mandrills is an old-world primate living in the very dense forest of Central Africa, mostly in Gabon. They form large multi-male, multi-female social groups, often composed of several hundreds of individuals. They reproduce seasonally and are sexually promiscuous. Males disperse from their natal group around puberty, and females live in their group from birth to death. Males are also transient members of this group. Some of them may spend only a few days in the group, others a few months to a few years. Mandrills form a typical matrilineal society where maternally related females and young males show highly differentiated social relationships. So I have depicted here 3 matrilines from one given social group. So because maternally related mandrills live in the same social environment since their birth, it seems parsimonious to assume that familiarity alone may serve individuals to recognize each other as maternal kin. So we will come back later on these mechanisms of kin recognition. So in mandrills, reproduction is highly skewed towards the alpha male, so in black here, who size every year about 60 to 70% of all offspring. So each year, each matriline therefore contains newborns that are related through the paternal line, black babies here on this graph, sized by the alpha male. So these black babies are in large majority in the group, and they are also found in every matriline. So these paternal alpha sibs are, however, not familiar or less familiar to each other than maternal alpha sibs are, because most of them are raised in different maternal or matrilineal environments. So in the study group of mandrills, we have recently estimated that these paternal alpha sibs, so PHS, and you will see this acronym all along the talk, okay? PHS for paternal alpha sibs, they are on average 2.7 times more numerous than maternal alpha sibs or MHS in the group. However, the variance in the number of paternal alpha sibs available to interact with is more than 30 times greater than the number of maternal alpha sibs available. So that means that each given newborn will either have many paternal alpha sibs present in its group or very few, especially when subordinate males side them. So this social framework that mandrills experience, as probably in other species too, made us to predict that a performant recognition system should have evolved to accurately recognize and socially discriminate paternal alpha sibs because of the high indirect benefits that they may enjoy, as predicted by the kin selection theory. So my talk will be organized as follows. First, I will show you some evidence in primates of differentiated social relationships among paternal alpha sibs. Then I will talk about the mechanisms of kin recognition in mandrill with some new results this time. And finally, I will present very briefly a few preliminary results on the adaptive value of social relationships. So before I started my PhD, there were only a few papers in the primate literature showing evidence of differentiated social relationships among paternal kin in 3 main social contexts: paternal care and incest avoidance, 2 contexts that do not involve kin selection processes, but probably involve similar recognition mechanisms, and nepotism. So in yellow baboons, Susan Alberts was the first to show that paternal alpha sibs copulate less with each other during consortships than other sexual pairs. Age similarity was proposed as a likely mechanism of recognition because paternal alpha sibs tend to have similar ages in baboons. In rhesus macaques, paternal sisters also are more nepotistic to each other than non-kin pairs. And again, age similarity among these kin probably explains this observation. More recently, marine accords showed in blue monkeys similar results on increased nepotism among paternal sisters, but this time similarity in age did not explain those results, and Marina proposed that phenotype matching should be a likely mechanism. So I will again come back later to this mechanism. Okay, so this was in this scientific context that I tried to replicate some of these findings during my PhD and later, both on captive and then on wild mandrills. So during my PhD, that occurred a very long time ago, unfortunately, I established the full pedigree of a semi-free ranging colony of mandrills housed in large forested enclosures at CIRM, a medical research center, also located in southern Gabon. I also made behavioral observation on 3 cohorts of juveniles for more than 2 years. So I go very fast with these results because, as I said, they are kind of old. So as for baboons, I generated an affiliation index based on grooming and spatial association between 22 juveniles and their groupmates. So here I'm showing you results on affiliation between those juveniles and the adult female of their group, including their mother, their maternal and paternal half siblings, and their non-kin female. Okay. So while controlling for several factors, I showed that mom offspring displayed the highest affiliation index as expected, but also that maternal and paternal half siblings both affiliate more with each other than non-kin pairs. So surprisingly, I did not observe any difference in the affiliation index between maternal and paternal half siblings, while in other primates studied so far, maternal kin often show higher nepotism than paternal kin do. So way later, I was able to replicate some of these findings, but this time in wild mandrills. So we recently investigated social relationships among 45 adult females from the natural population for whom we had the genetic ancestry. So on this graph, I'm showing you the raw frequencies of grooming, aggression, and spatial association among paternal half sisters in red, maternal half sisters in orange, and non-kin pairs of females in yellow. So what we can see from this graph is that maternal and paternal half sisters, okay, tend to show similar social phenotypes. They groom and aggress each other more than non-kin pairs do, and they are also spatially closer, although this last analysis did not reach significance. So I hope that now it's kind of clear to you that several primates do bias their behavior towards their paternal half siblings, even if they are unfamiliar or less familiar with each other. They are not living in the same maternal environments. So the next step was just to explore the mechanisms of kin recognition, and again, of course, in mandrills. So in 2007, Andrea Vidig wrote a review on paternal recognition mechanisms. So 3 main hypotheses emerged, one based on age similarity, as I said before, because very often paternal half siblings are close in age, as males' tenure is generally restricting in length. So Anya further proposed that some forms of familiarity may develop among paternal half siblings to facilitate recognition. For example, if females associate with the males they mated with following their offspring birth, then these offspring, born from different females but sired by the same father, could thus be secondarily associated since their birth, creating a familiar environment, social environment. The third mechanism, the so-called phenotype matching, assumes that phenotypic similarities correlate with genotypic similarities. This mechanism supposes that an individual knows somehow its own phenotypic attributes or those of non-kin to match them with those of potential kin to assess relativeness. So at the time of this review, phenotype matching was virtually untested in large mammals, mainly because it's hard to disentangle from familiarity processes. So I've been exploring this mechanism for the past few years, thanks to a unique setting that I'm going to present you now. So if you remember, I told you at the beginning of this talk that the natural population of mandrills that I'm studying originating from a captive colony housed at CIRM. So throughout the years, individuals from both populations continue to reproduce, generating a situation where some individuals from these two populations are related to each other, but are also totally unfamiliar because they never lived together. So in collaboration with Florence Levrero, as you can see on this picture, from the University of Saint-Etienne, we started collecting vocalizations on both populations to study first whether a genetic signature was detectable in Mandrill's voices. So to do so, we studied their most common call, a contact call. So this is it, and this is the corresponding sonogram. Okay. So Florence extracted 7 acoustic parameters to generate acoustic distances across pairs of individuals. So these acoustic distances on the y-axis of this first graph contains signal of relatedness, but in combination with a degree of familiarity of the studied pairs. So 3 classes of genetic relatedness are plotted on this figure for clarity, with highly related individuals located on the right of the graph. So across all types of dyads in light gray, or only across unfamiliar dyads in dark gray. So basically, the take-home message of this first graph is that highly related dyads show more similar voices, or are less distant acoustically. And this relatedness effect seems to be even stronger among unfamiliar dyads that are in dark gray. So when splitting these studied pairs in 3 kin classes, non-kin, NK, paternal or maternal half-sib, PHS and MHS, we showed that these 3 classes significantly differed from each other, with non-kin showing less similar calls than either maternal or paternal half-sibs. However, these 2 latter categories also differ as maternal kin appeared more similar than paternal kin are, although both categories share on average the same degree of genetic relatedness. So because maternal half-sibs live in the same matrilineal environment, we supposed that their familiarity further impacts their voices. And this last graph confirms this point. Pairs that never live together, so on the left of the second graph, okay, show very different contact calls, but the longer they live together, up to 2 years or more than 2 years, 6 years, sorry, the closer their voice were. And whatever their degree of genetic relatedness, in dark gray, for example, we have only depicted unrelated pairs. So this set of results indicates that genetic relatedness does impact Mandrill's contact calls, but that these calls are flexible because familiarity further change their structure. So following that, those results, we asked whether this genetic signature found in contact calls may help Mandrills to discriminate their unfamiliar kin, using therefore a mechanism of phenotype matching of voices. So the unique setting of the 2 studied populations allowed to perform playback experiments on the captive colony with contact calls sampled in the wild population from unrelated, unfamiliar, or familiar conspecifics. So while we were able to study the impact of genetic relatedness on responses to playbacks, we were unable to discriminate between different classes of kinship as before because of a limited sample size. Oops, sorry. What did I do? Yeah. So when controlling for familiarity, so unfamiliar dyads are shown in dark gray here on this graph, we demonstrated that individuals that heard voices of kin behaviorally respond more than to voices of unrelated mandrills. So this graph represents the number of movements performed towards the speaker as a function of the genetic relatedness between the producer of the voice and the receiver. So yes, phenotype matching occurs in mandrills, or seems to occur in mandrills. Individuals do not have to know their relatives to behaviorally discriminate them, although this is modulated by the degree of familiarity of the dyads, as the social environment impacts the structure of voices too. So, sorry, I'm drinking. So more recently, I have started new studies based on face recognition. So it comes to me when I was in the field watching the study population of wild mandrills. So there were these 2 individuals, so an adult female on the right here and an adolescent male on the left, that resembled a lot to each other, which was kind of surprising given their sexual size dimorphism. And when looking at their pedigree, I realized that they were sired by the same father. They were very different in age, actually. So it seems perhaps a bit silly to you, but this is how everything started. So I have a little game for you, although I'm not sure this is a good idea through Zoom, but you can guess in your head, and then you will tell me later at the end of the talk if you are right or not. So you have here 4 adult females. So the focal at the top and 3, the focal, sorry, an adult female focal at the top, and 3 of her social partners at the bottom. One is her maternal half-sister, another a paternal half-sister, and a third one is a non-related adult female. So guess who is who? So I give you a little time. And here is the answer. So to me, at least to me, it seems pretty obvious that these 2 paternal half-sisters look way more to each other than the 2 other pairs. So you will let me know if you agree or not with that. So these naturalistic observations led us to ask the following question. Does kin-biased facial resemblance merely reflect genetic resemblance or result from kin selection processes to improve kin recognition and behavioral discrimination? So if our hypothesis is true, then paternal half-sibs should resemble more to each other than predicted by their genetic relatedness alone. So they should be more facially similar than maternal half-sibs are, okay? We took advantage of the large photo bank we had collected throughout the year and used a powerful approach based on artificial intelligence methods and the training of deep neural networks to generate facial distances across pairs of individuals. So this was done in collaboration with Julien Renaud, a CNRS colleague of mine and also my friend. So artificial intelligence methods are very powerful, necessitate large training data sets that may be based on a few individuals only, but with hundreds of portraits. So the long-term accumulation, storage, and treatment of pictures collected on the study population just allowed that. So we generated facial distances across 38 adult females with a fully solved pedigree and first showed a negative, significant relationship between facial distance, here on the y-axis, and genetic relatedness of the studied pairs of females on the x-axis of this graph. So on this figure, the average face distance across pictures taken from the same individuals is also displayed with a white diamond here to give you an idea of the intra-individual resemblance across pictures, okay? We were interested, however, to compare maternal to paternal half-sisters, but both categories roughly fall within this range of relatedness. So I remind you that our initial prediction was that if facial resemblance is under kin selection, paternal half-sisters should resemble more to each other than expected given their genetic resemblance. So we splitted pairs of adult females, so here it's an adult female, into 3 kin classes. So on this graph, paternal half-sisters are shown in red, maternal half-sisters in orange, and non-kin pairs again in yellow. So as you can see, there is a significant effect of kinship on face distance on the y-axis, but in combination with the age difference of the studied pairs of pictures of adult females. When this age difference is less than 2 years, paternal half-sisters in red resemble more to each other than maternal half-sisters do, and both more than non-related females in yellow. So when the age difference is greater than 2 years, then the difference between paternal and maternal half-sisters fades. We further analyzed face distance across juvenile females, as on this picture, and found globally the same effect as for adults. At these young ages, females are between 2 and 4 years old. Paternal half-sisters resemble more to each other than maternal half-sisters do, who this time appear not different from non-kin. Okay, there is no significant difference between orange and yellow females, as you can see here. This effect is also particularly true when the difference in age was less than a year, although the same pattern remains for juvenile females that are more than a year apart. So to conclude, from juvenility to adulthood, paternal half-sisters resemble more to each other than maternal half-sisters. So we propose that this increased facial similarity evolved, or probably evolved, among paternally related females to improve kin recognition. Okay, to sum up, we know now that paternal sisters resemble more to each other than expected. We also know that they show differentiated social relationships. So discrimination of paternal sisters therefore probably occurs through the evaluation of facial traits. So how do mandrills do? How do they know? How do mandrills know what they look like exactly? So I doubt that young mandrills have a good idea of their face, right? Probably precluding a mechanism of self-referent phenotype matching. So we propose that this increased facial resemblance across paternal alphabets may be evaluated and used by third parties to influence patterns of association between them. That's what we have recently tested in the studied species in mandrills. So because paternal resemblance arises at very young ages, as I showed, and that mandrill fathers are half of the time absent from their offspring group at their birth, we propose mothers as good candidates to mediate the relationship of their own offspring with their paternal alphabets. We further expected this to happen very early in life when offspring are still highly dependent on their mothers. So we use a dataset composed of portraits taken on 80 infant mandrills. So unfortunately, and because genetic analyses are challenging to do in the field, especially on the infants, paternity was determined for only 15 of them. So what we did is that we considered those infants conceived during the same tenure of a given alpha male and compared their facial resemblance to those infants that were conceived during different alpha males' tenure. So controlling for infants' age and sex, as well as their maternal identity, we found that those infants born during the tenure of the same alpha male or that were sired by the same genetic father resemble each other significantly more, so on the left, right, than those infants that were born to different fathers. So in contrast, at these young ages, maternal alphabets do not look more alike than infants born to different mothers. So these results therefore strongly supports and extends one of our previous results. Starting as early as infancy and continuing throughout juvenescence and adulthood, paternal alphabets resemble each other more than maternal alphabets do. And this is a very strong signal that we found. So increased facial resemblance among paternally related mandrills calls for an adaptive explanation involving phenotype matching. So as I said earlier, we hypothesize that mothers use this phenotypic information to foster their offspring social opportunities, deriving fitness benefits through an increase in offspring fitness. So we use pictures from infants for which we had detailed records on spatial association during their first year of life. Okay. So I'm going to show you 3 similar graphs now on the association between mothers and other infants, among infants, and between mothers of 2 infants as a function of the facial distance between these infants. So I hope I'm clear. I'm sorry if I'm not. So we control for different confounding variables in blue, but I won't talk about them today. So this graph represents the estimate of the spatial association between mothers and other infants, with more associated pairs being on the right, okay, of the graph. So we showed that mothers are less close to babies that do not resemble their own offspring. In other words, mothers are spatially closer to stranger babies that resemble more their own, as we earlier proposed. So here is a similar graph, but based on association recorded among infants, again, as a function of their facial distance in purple. So infants are also less associated with when they do not resemble to each other. So young mandrills associate more with similarly looking peer mates. Finally, this third graph represents the association among mothers as a function of the facial distance of their babies. So as you can see, there is no such significant relationship between both variables. Mothers thus actively associate with similarly looking infants, but not with their mothers. So by doing so, mothers may thus influence their offspring's social preferences towards similarly looking other infants that are also more likely to be paternal alphabets. At these young ages, infants highly follow what their mother do. So this high directionality of mother-offspring relationships thus strongly suggests that mothers mediate association patterns among infants. These results, again, call for an evolutionary explanation because paternal kin share paternal, not maternal genes. So it seems thus essential to explore the fitness benefits that mothers may derive from favoring nepotism between their offspring and their paternal kin. So because I needed to formalize that, I started a collaboration with a theoretician from my lab, François Rousset, as you can see here. So François wrote many equations that can be summarized on this very simple figure. So the face at the top represents a given actor, for example, a focal mother, who may derive direct fitness benefit through her action on her offspring or indirect fitness benefit through her action on her kin. So this is a very simple representation of the kin selection framework, okay? So with François, we showed that under minimal assumptions, mothers can also obtain fitness benefit by fostering interaction between their offspring and their paternally related siblings, a process here in red that we call the second order kin selection. So crucially, this selection process does not imply the infant's social partner, the red face here, to be related to the mother. It's therefore distinct from classic kin selection. So by favoring nepotism between their own offspring and their paternal half-siblings, mother can thus derive direct fitness benefits. And this is the scenario A here. But this mechanism described can be further generalized to any actor whose behavior promotes positive interaction between a non-kin recipient, still in red, and any actor's kin. And this is the scenario B that I have depicted here. So while the second order kin selection process is a probably novel explanation for the evolution of nepotism, and more generally, for the behavioral discrimination of kin, it perfectly fits into the mathematical framework offered by the inclusive fitness theory, as you can see on this very, very simple figure. So now very quickly, a few words about the adaptive values of social relationship in social species. So the benefits, sorry, it's a bit different from the two other parts, but I wanted to talk about it a bit. So the benefits of differentiated social relationships, such as increased affiliation or proximity, can affect health, immunity, reproduction, and survival in multiple species, including in human, in non-human primates, and also in other vertebrates. So for example, female baboons that are socially better integrated enjoy higher offspring survival and live themselves longer. In summary-free ranging mandrills that I studied during my PhD, those females that are better socially integrating during their juvenile period, measured as the cumulated affiliation exchange with their group mates on the x-axis of this graph, also enjoy a decrease in their age at first birth of about a year compared to those females that are poorly socially integrated. So as for baboons, though, this effect was true when considering all of their social partners, not only their close kin. Recently, and in collaboration with Sébastien Sosa and Cédric Sueur from the University of Strasbourg, we have used a social network approach to study the relationship between several measures of social integration of 60 female mandrills based on their grooming relationships, their association, and their cortisol levels, using, again, long-term data on behavior and physiology that we have accumulated. So preliminary results show that cortisol level, FCM, on this graph decrease with the strength of social relationships, but increase with the number of social partners. What seems valuable to female mandrills is thus to have a few social partners, but with strong social bonds, a result which is also reported in other primates. So a next step that I would like now to take is to evaluate whether social choices vary in their adaptive value. A recent study on grooming networks in rhesus macaque indicated that highly central females within their family network had lower cortisol levels than females with similar high centrality, but with non-established group mates. So the same type of behavioral interaction, so here grooming in macaques, seems thus to have different functions and even opposite effects on health, depending on the social context. So I'm done for today, and I hope my English was clear enough for you to follow. So I'd like to thank you for listening to me, and I'm also very grateful to all of my past and present field assistants here with my daughter, who are daily dedicated to the study of wild and elusive mandrills. Thanks a lot.
Great, thanks a lot, Marie.
Yeah.
We have one question. People can type in more questions, but I'm going to start with a question, which is, what do we know about paternal care in mandrills, or if it's not yet known, what do you think about maternal care in mandrills?
So,
paternal care.
So we know not that much, but hopefully we will know better soon. The things that we know is that, so as I said, mandrills, male mandrills come for only short period of time or restricted period of time in the group, and half of them has left when their offspring are born. Okay, when they are here, we know that some male mandrills, not always the former alpha male, form like something that we call crèche. Do you understand that? Like kindergarten. So there is always one male in the group at the birthing season, which is followed by many infants. I think this is the same in baboons. So we are currently studying whether this male is likely to be the father, which is probably not always the case. And the second thing that I can say is that male mandrills do not carry their offspring or do not carry infants. They do not groom them at all. So sometimes they can perhaps protect them, or at least they are patrolling the group. So they intervene during conflicts. And sometimes infants are involved during those conflicts. And the last thing I want to say is that, so during my PhD, I studied a little bit association pattern between males and infants, and in general, the father is closer to their own infants than a non-genetic male. Okay?
Got it.
But we are currently studying it, and in particular, those crèches.
Cool. We have a couple of other questions. First from Mark Halber, who says, wonderful talk, thanks. In the acoustic phenotype matching that you described, do the offspring learn the mother's voice and match that against unfamiliar relatives, or do you think it's a self-referential phenotype matching?
That's a very interesting question. That's a very interesting question. So I don't know. Both could occur, definitively. And so now I'm trying to think of how to test that. Probably hard because infants, of course, they are always with their mother. So I'm not sure I can test that, but I'm sure that both mechanisms could occur, yeah.
And Sahas Parva says, amazing talk. Does the resemblance to fathers drive levels of matings between fathers and daughters? So does the facial resemblance facilitate inbreeding avoidance? Okay,
so most of the time, when the daughters are mature and are sexually mature, fathers are not here anymore. So the opportunity for incest to occur is probably very restricted in mandrills, okay, because they are staying only for a short period of time, those males. So, and it seems that there are inbreeding avoidance between fathers and daughters, yeah. Because males in captivity, stay in the group because they cannot, of course, emigrate from the group. So, yeah.
And then
I'm not. So I don't know if facial resemblance help or not. I don't know.
Got it. And then I think we have one final question from Liz.
Great talk. I was curious if we know anything about how these juvenile social relationships that might be mediated by the mother's matching between paternal kin and her offspring develop into adult relationships. So are these juvenile relationships translate into adulthood? Or we know anything?
I don't know yet.
Yeah.
Perhaps in 10 years. Ask me in 10 years.
Great, I will.
That answer has been a frequent refrain in the seminar series, and so we will ask in 10 years.
Okay. Okay.
Well, Marie and Nikos, thanks for two really interesting talks. It was fun to hear about the Mandrill project, especially some of the history and the things that you're doing there, and I'll look forward to hearing more in the future. So thanks so much for sharing with us.
Thank you. to you.
For everybody in attendance, we'll be here at the same time, same place next week. See you all then.
Okay, bye-bye. Thanks.
Bye-bye. Thanks,
Bye.
everybody.